Glutamate receptors, particularly AMPA and NMDA receptor subtypes, are crucial for excitatory neurotransmission and synaptic plasticity in the central nervous system. Their dynamic regulation and trafficking within neurons underpin key processes such as learning, memory, and adaptation to neural activity. At the synaptic membrane, glutamate receptor numbers and subunit composition are not static. Instead, they are tightly regulated through a balance of insertion, removal, recycling, and degradation. This trafficking is governed by a complex interplay of molecular signals, scaffolding proteins, and cytoskeletal elements. AMPA receptors, AMPARs, are especially mobile. Under basal conditions, a pool of AMPARs cycles between intracellular compartments and the synaptic membrane. During synaptic potentiation, such as long-term potentiation, LTP, there is an increased delivery of AMPARs to the postsynaptic density, enhancing synaptic strength. Conversely, long-term depression, LTD, triggers internalization and removal of AMPARs, weakening synaptic transmission. These processes involve post-translational modifications, like phosphorylation, interactions with PDZ domain-containing scaffold proteins, PSD95, and actin cytoskeleton remodeling. NMDA receptors, NMDRs, also undergo regulated trafficking, though their surface expression is generally more stable than that of AMPARs. NMDAR trafficking is influenced by developmental stage, neuronal activity, and subunit composition, which affects receptor localization and function. The dynamic exchange of NMDAR subunits at synapses can alter calcium permeability and downstream signaling pathways. Endocytic and exocytic pathways mediate the internalization and reinsertion of glutamate receptors. Clathrin-mediated endocytosis is a major route for receptor removal, while recycling endosomes facilitate their return to the membrane. Ubiquitination and degradation via the lysosomal or proteasomal pathways ensure the turnover of receptors that are no longer needed. Activity-dependent changes in glutamate receptor trafficking provide a rapid and reversible means for neurons to modulate synaptic efficacy. Disruptions in these processes have been implicated in various neurological disorders, including epilepsy, schizophrenia, and neurodegenerative diseases, highlighting the importance of precise regulation of glutamate receptor dynamics in maintaining healthy brain function.